What if the difference between people who develop aggressive cancer and those who never do has less to do with their genes and far more to do with what ends up on their plate every day? Most people grow up believing cancer is purely written in DNA, that once damage happens, it cannot be reversed, and that nutrition plays almost no meaningful role once a cell turns cancerous. But after nearly 15 years working in emergency medicine and seeing oncology patients up close, I've learned something that doesn't fully align with what many of us were taught in medical training. There are certain foods that appear to support the body in repairing DNA damage and, even more surprisingly, may help restrict the growth of tumors by cutting off their blood supply. Not just slowing them down, but essentially starving them of what they need to expand. Because at a microscopic level, a tumor without blood vessels cannot grow beyond a tiny size. It stays almost frozen, but once blood vessels connect to it, the situation changes dramatically and growth can accelerate in a very short period of time. My name is Dr. Becker, and I work in emergency medicine. Over the years, while treating patients with cancer, I noticed a recurring gap between clinical treatment and nutritional discussion. Patients were often told that food doesn't really matter in this context. But when I looked deeper into the research, I found studies that have existed for decades suggesting that certain foods can activate natural protective systems inside the human body, systems that are directly involved in fighting abnormal cell growth. Yet in everyday practice, this information is rarely discussed. In this video, I want to break down what the science actually shows. We will look at how specific foods may influence DNA repair at the cellular level and how the body can potentially limit cancer growth by controlling a process called blood vessel formation. We will also explore why this process is so central to how tumors develop and how nutrition may influence it in measurable ways. Before going deeper, there is something important to understand about cancer itself. Cancer cells are not foreign invaders that suddenly appear from nowhere. In fact, abnormal cells are being formed in every human body all the time, with around 40 trillion cells constantly dividing and replacing old ones. Copying errors are unavoidable. It's similar to photocopying the same document repeatedly. Eventually, small mistakes begin to appear. Some of these mistakes result in abnormal cells, and a small number of those can become cancerous. The surprising part is that most people already have microscopic clusters of such cells forming and disappearing throughout life. This is not unusual or immediately dangerous. The reason most of us do not develop cancer every day is because the body has surveillance systems in place. The immune system constantly scans for abnormal cells and removes them before they can spread. It acts like a security force that identifies and clears potential threats before they grow into something harmful. However, even if some abnormal cells manage to escape detection, they still face a major limitation. They cannot grow without resources. For a tumor to expand, it needs oxygen, nutrients, and energy. And to access those, it requires blood vessels. This is where a biological process called angiogenesis becomes critical. Angiogenesis simply refers to the formation of new blood vessels. The body carefully regulates this process because it must remain in balance. Too little blood vessel growth leads to tissue damage. Too much can create problems as well. You can think of it like maintaining a lawn at the right height. The system constantly trims when things grow too much and allows growth when needed. This balance keeps everything stable under normal conditions. But cancer cells have a way of interfering with this system. They can release chemical signals that essentially trick the body into growing new blood vessels toward them. Once that happens, a previously small and contained cluster of abnormal cells suddenly gains access to oxygen and nutrients. At that point, growth can accelerate rapidly, turning something microscopic into a much larger mass in a very short time. Understanding this shift is essential because it sets the foundation for everything that follows. The key question is not only how cancer starts, but what allows it to grow. And that brings us directly into the science of how blood vessel formation can be influenced and why nutrition may play a role in that process. In the next part, we will go deeper into how tumors exploit angiogenesis, what happens when blood supply reaches them, and why controlling this process has become one of the most important areas in modern cancer research. To understand why cancer becomes dangerous, we need to focus on what changes the moment a tumor gains access to blood vessels. Before that point, even abnormal cells remain limited. They exist in a restricted state, unable to expand beyond a very small size because they simply cannot get enough oxygen or nutrients. In many cases, they can remain dormant for years without causing any noticeable harm. But the moment blood vessels reach them, everything changes. This process is called angiogenesis, and it is one of the most important biological switches in cancer development. When new vessels begin growing into a tumor, it is no longer isolated. It becomes connected to the body's full supply network. That connection allows it to grow, divide, and spread at a dramatically faster rate. To put it into perspective, research has shown that an isolated tumor without blood supply typically remains extremely small, often around 1 to 2 millimeters in size. At this stage, it is so small that it can remain unnoticed for a long time and may never progress further. It is essentially contained. However, once angiogenesis begins and blood vessels penetrate the tumor, the behavior changes completely. Studies have observed that tumor growth can increase exponentially after vascularization occurs. In some documented cases, growth rates can multiply thousands of times within a short period, transforming a previously dormant cluster into a rapidly expanding mass in a matter of days or weeks. This is not a slow, linear process. It is a biological acceleration triggered by access to resources. Because of this, angiogenesis has become one of the most studied areas in cancer research. Scientists have long recognized that if blood vessel growth toward tumors could be controlled, it might be possible to limit cancer progression itself. This has led to the development of pharmaceutical treatments known as angiogenesis inhibitors. These drugs are designed to block the signals that tumors use to attract new blood vessels. In clinical settings, some of these treatments have shown effectiveness, particularly in slowing tumor growth. However, they also come with limitations and side effects, largely because they do not distinguish between harmful and healthy blood vessel formation. The body still needs angiogenesis for normal processes like healing and tissue repair, so interfering with it system-wide is complex. What became even more interesting over time is that this same biological pathway is not only influenced by drugs, it can also be influenced by compounds found in everyday foods. When researchers began testing natural plant compounds using laboratory models designed to measure blood vessel growth, they made a surprising discovery. Certain food-derived substances were able to reduce or inhibit angiogenesis in controlled environments. These were not synthetic chemicals, but natural compounds extracted from plants and common dietary sources. One of the earliest and most well-documented examples came from green tea. Scientists isolated a compound called EGCG, a type of polyphenol found in tea leaves. In experimental models, EGCG showed the ability to interfere with signals that promote blood vessel formation. In some cases, its effects were strong enough that researchers noted it behaved similarly to compounds being developed in pharmaceutical research. This does not mean green tea is a treatment for cancer, but it does highlight something important. Biological pathways involved in cancer are not isolated from nutrition. They respond to chemical signals, and some of those signals come directly from food. As research expanded, scientists identified that green tea was not unique. When a wide range of foods were tested under similar laboratory conditions, over 100 different foods showed measurable effects on angiogenesis-related pathways. These foods contain compounds like flavonoids and polyphenols that interacted with the same biological mechanisms tumors use for growth. This shifted the perspective from viewing food as neutral to understanding it as biologically active at the cellular level. At the same time, another critical mechanism came into focus: DNA repair. While angiogenesis controls whether a tumor can grow, DNA repair determines whether abnormal cells form in the first place. Every day, cells in the body experience damage from environmental exposure, internal metabolic activity, and external stressors. Under normal conditions, the body has built-in repair systems that fix this damage before it becomes permanent. But these systems depend on specific nutrients to function properly. When those nutrients are insufficient, damage can accumulate over time, increasing the risk of mutations. Think of it like infrastructure maintenance. A city road system can handle wear and tear if it has a functioning repair crew and materials. Small cracks get fixed immediately. But if resources are lacking, those cracks expand, multiply, and eventually lead to structural failure. DNA works in a similar way at the cellular level. This is where certain foods begin to show measurable effects in research. One of the most surprising findings came from studies involving kiwi fruit. In controlled trials, participants who consumed kiwi daily showed significant reductions in markers of DNA damage. Even more interesting, higher intake was associated with improved DNA repair activity within cells. The mechanism appears to be linked to vitamin C and a range of antioxidant compounds found naturally in kiwi. These compounds help neutralize reactive molecules that damage DNA strands, while also supporting enzymatic systems responsible for repair. What makes this especially important is not just the presence of vitamin C, but the combination of multiple compounds working together. Whole foods provide a complex mix of nutrients that interact in ways isolated supplements often cannot replicate. As research continued, a clearer picture began to emerge. Cancer development is not driven by a single factor. It is influenced by multiple biological systems working together, including immune surveillance, angiogenesis, DNA repair, and metabolic regulation. And increasingly, evidence suggests that these systems are sensitive to dietary inputs. In the next part, we will explore how DNA damage occurs on a daily basis, how foods like kiwi directly influence repair mechanisms, and why nutrition may play a far larger role in cellular protection than most people realize. If we zoom in even further into what is happening inside the body every single day, a more detailed picture starts to form. Cancer does not begin as a sudden event. It develops gradually through accumulated damage at the cellular level, much of which is happening constantly, even in healthy individuals. Every cell in the body contains DNA, and that DNA is under continuous pressure. It is exposed to internal metabolic byproducts, environmental toxins, radiation from sunlight, and even normal biological processes that generate unstable molecules. These molecules can interact with DNA and create small breaks or mutations. Most of the time, this is not a problem because the body is equipped with repair systems. These systems act like maintenance crews, constantly scanning DNA for damage and fixing it before it becomes permanent. However, this repair process is not automatic in the sense that it runs independently of resources. It depends heavily on nutrients and biochemical support. When the body has what it needs, these repair mechanisms are highly efficient. Damaged sections of DNA are quickly corrected and cells continue functioning normally. But when nutritional support is lacking, or when oxidative stress is too high, the repair systems can become less effective. Over time, small errors can accumulate, and this is where long-term risk begins to increase. To understand this better, think of DNA like a massive library of instructions. If a few letters in a book are damaged, the meaning might still be understood, and repairs can be made easily. But if damage keeps accumulating without repair, entire instructions can become corrupted. That is when cells begin to behave abnormally. This is why researchers have become increasingly interested in foods that support DNA stability and repair. One of the most well-documented examples comes from studies involving kiwi fruit, which produced results that surprised even many scientists in the field. In controlled clinical research, participants were given varying amounts of kiwi daily over several weeks. What made these studies particularly interesting was not just whether damage decreased, but whether the body's repair capacity actually improved. At the end of the study period, researchers used specialized laboratory techniques to measure DNA damage in blood cells. The results showed a clear reduction in DNA strand breaks among those consuming kiwi regularly. Even more notable was that higher consumption levels were associated not only with reduced damage, but with enhanced repair activity. This suggests that kiwi is not just preventing new damage, but actively supporting the systems that correct existing damage. The primary mechanism appears to involve vitamin C, a well-known antioxidant. Vitamin C plays a direct role in neutralizing reactive oxygen species, which are unstable molecules that can damage DNA. By reducing these reactive compounds, vitamin C lowers the overall burden on the repair system. But kiwi contains more than just vitamin C. It also provides a wide range of polyphenols, flavonoids, and other micronutrients that appear to work together in a synergistic way. This synergy is important because the body rarely relies on single compounds in isolation. Biological systems function through networks of interacting nutrients. This is where whole foods differ significantly from isolated supplements. A vitamin C tablet delivers one compound. A kiwi delivers that same compound alongside dozens of others that may influence absorption, stability, and effectiveness. The combined effect can be greater than the sum of its parts. As research expanded into other foods, scientists began to see similar patterns. Certain berries, green tea, cruciferous vegetables, and legumes all showed measurable effects on cellular protection pathways. These foods were not acting through one single mechanism, but through multiple overlapping systems. Some influenced oxidative stress. Others affected inflammation. Others appeared to modulate gene expression or immune signaling. Together, they contribute to a more stable internal environment where DNA damage is less likely to accumulate. At the same time, another important layer of protection exists in the immune system. Even when abnormal cells do form, the body has mechanisms to detect and remove them before they become dangerous. Specialized immune cells continuously patrol tissues, identifying cells that behave abnormally and eliminating them. However, the effectiveness of this surveillance system can vary depending on multiple factors, including nutrition, gut health, inflammation levels, and overall metabolic state. This is where diet again becomes relevant, not as a cure, but as a supporting factor that influences how well these systems function. Emerging research has also shown that gut bacteria play a surprisingly important role in this process. The microbiome is not just involved in digestion, it also interacts with immune function and inflammation regulation. Certain bacterial strains are associated with stronger immune responses and better outcomes in disease prevention contexts. This connection between gut health and immune surveillance adds another layer to the relationship between food and cancer risk. It is not just about one nutrient or one mechanism, but about how diet shapes multiple biological systems simultaneously. As we continue building this picture, it becomes clear that cancer development is influenced by a balance between damage and repair, growth and restriction, detection and elimination. When this balance is maintained, abnormal cells are less likely to progress. When it is disrupted, risk increases. And importantly, many of the systems involved in maintaining this balance are sensitive to daily nutritional inputs. In the next part, we will look more closely at how specific foods influence immune activity, how compounds like anthocyanins and flavonoids affect immune cell function, and why some dietary patterns are associated with stronger cancer surveillance at the cellular level. Once we understand how DNA damage and repair work, the next logical question is what happens when abnormal cells manage to escape these early defense systems? Because even in a well-functioning body, not every abnormal cell is eliminated immediately. Some can slip past immune detection, and others can persist in a dormant state for long periods. This is where the immune system becomes critical again, not just as a backup mechanism, but as a continuous surveillance network that operates every second of the day. Specialized immune cells circulate through the bloodstream and tissues, constantly scanning for signs of abnormal behavior. If a cell appears damaged, unstable, or cancer-like, these immune cells can trigger its destruction before it becomes dangerous. Among the most important of these are natural killer cells and T cells. Natural killer cells act quickly and do not require prior exposure to a threat. They can identify abnormal cells based on surface markers and eliminate them immediately. T cells on the other hand, are more specialized and adaptive, capable of targeting specific abnormal patterns over time. The strength of this immune surveillance system is not fixed. It can be influenced by many factors, including sleep, stress, physical activity, and importantly, nutrition. This is where dietary compounds begin to show measurable effects on immune activity. One of the most studied examples comes from blueberries. These small fruits contain compounds called anthocyanins, which are responsible for their deep blue color. Beyond their appearance, these compounds have been shown to influence immune function in meaningful ways. In controlled studies involving human participants, regular consumption of blueberries was associated with increased activity of immune cells involved in cellular defense. In some cases, researchers observed significant increases in circulating T cells after consistent intake over several weeks. This was not a minor fluctuation, but a measurable shift in immune activity. The mechanism appears to involve both antioxidant effects and direct signaling pathways that influence immune cell behavior. Anthocyanins reduce oxidative stress, which can impair immune function, while also supporting communication between immune cells and other systems in the body. What makes this especially interesting is that immune enhancement does not necessarily mean overstimulation. Instead, these compounds appear to support balance, helping the immune system respond more effectively without becoming excessively active. At the same time, inflammation plays a central role in how the immune system behaves. Chronic low-grade inflammation can interfere with immune surveillance and create an environment where abnormal cells are more likely to persist. Many plant-based compounds help regulate this inflammation, reducing unnecessary immune burden while preserving essential defense activity. Another important layer of immune function involves the gut microbiome. The human digestive system contains trillions of bacteria that interact closely with immune cells. These microbes are not passive residents. They actively influence immune signaling, inflammation levels, and even how the body responds to disease. One particular bacterial species that has gained attention in recent research is Akkermansia mucinifera. Studies have found that individuals with higher levels of this bacterium often show stronger immune responses in certain clinical contexts. In cancer research, its presence has been linked to improved outcomes in patients undergoing immunotherapy. Immunotherapy is a treatment approach that helps the immune system recognize and attack cancer cells more effectively. However, not all patients respond equally. Research has shown that gut microbiome composition can be one of the key factors influencing this response. In one major study, scientists analyzed cancer patients receiving immunotherapy and discovered a striking pattern. Those who responded well to treatment consistently had higher levels of beneficial gut bacteria, including Akkermansia. Those who did not respond often lacked these microbial populations. This suggests that the gut microbiome plays a role in priming to the immune system, essentially preparing it to respond more effectively to abnormal cells. Diet again becomes the primary driver of this system. Foods rich in fiber, polyphenols, and prebiotics help nourish beneficial bacteria, while highly processed diets tend to reduce microbial diversity. Over time, these dietary patterns can influence immune efficiency at a systemic level. At this stage, a clearer picture begins to form. Cancer prevention is not dependent on one mechanism alone. It involves DNA stability, immune surveillance, inflammation control, and microbial balance working together in coordination. And while genetics certainly play a role in susceptibility, they do not act in isolation. The environment inside the body, shaped significantly by daily lifestyle and dietary choices, determines how those genes are expressed and how effectively protective systems function. This does not mean food is a cure or replacement for medical treatment. Rather, it highlights that nutrition interacts with biology in ways that can influence the internal conditions under which disease develops or is controlled. In the next part, we will move from the science into practical application. We will break down specific foods that have been studied for their effects on angiogenesis, immune function, and DNA repair, and how they can be incorporated into daily routines in a realistic and consistent way. Now that we understand how the immune system, DNA repair, and inflammation all connect, the next step is to look at what this actually means in daily life, because science alone is not enough. If it cannot be translated into practical choices people can realistically maintain, the key question becomes which foods have been studied for their ability to influence these biological systems, and how can they be used in a consistent way without turning life into a restrictive routine. One of the most researched categories is foods that influence angiogenesis, the process that controls blood vessel growth. As discussed earlier, tumors depend on blood vessels to grow beyond a very small size. Without that supply, they remain limited. This is why researchers have spent decades studying compounds that can interrupt this process. Green tea is one of the most widely studied examples. It contains a compound called EGCG, a type of polyphenol that has been shown in laboratory settings to interfere with signals involved in blood vessel formation. In controlled experiments, EGCG can reduce the activity of pathways that tumors use to attract new blood vessels. This does not mean green tea is a treatment, but it does demonstrate that natural compounds can interact with the same biological systems that cancer depends on for growth. Beyond green tea, researchers have identified many other plant-based foods with similar properties. When tested under laboratory conditions designed to measure angiogenesis, a surprisingly large number of foods show measurable effects. These include foods rich in flavonoids, polyphenols, and other bioactive compounds. Another important category involves immune-supporting foods. Blueberries are a strong example due to their anthocyanin content. These compounds have been associated with increased activity of immune cells that target abnormal cells. In multiple studies, consistent consumption of berries has been linked with improved markers of immune function. What is particularly important here is consistency. The immune system does not respond strongly to occasional intake. It responds to patterns over time. Regular exposure to supportive nutrients creates a stable environment where immune cells can function more efficiently. Soy-based foods also appear frequently in population research. Despite ongoing misconceptions, soy contains compounds called isoflavones, including genistein, which have been studied for their interaction with hormone-related pathways and angiogenesis. In laboratory models, genistein has shown the ability to influence signaling pathways involved in blood vessel formation. Population studies have also observed differences in cancer rates between regions with high and low soy consumption. While many factors contribute to these differences, diet is considered one of the variables of interest. Cruciferous vegetables, particularly broccoli and broccoli sprouts, are another major focus of research. These foods contain sulforaphane, a compound that activates detoxification enzymes in the body. These enzymes help process and eliminate potentially harmful substances before they can cause cellular damage. Broccoli sprouts are especially concentrated sources of this compound. Even small amounts contain significantly higher levels of sulforaphane compared to mature broccoli. In experimental studies, sulforaphane has been shown to influence pathways related to cellular defense and detoxification. Tomatoes, particularly when cooked, provide another important compound called lycopene. Lycopene is a carotenoid that has been studied for its antioxidant properties and its role in protecting cells from oxidative stress. Interestingly, cooking tomatoes increases the availability of lycopene, making processed tomato products more bioavailable than raw tomatoes. Population studies have associated higher tomato intake with reduced risk of certain cancers, particularly when consumed regularly over long periods. The effect appears stronger when tomatoes are consumed with healthy fats, which improve absorption. Pomegranate is another food of interest due to its L-gitanin content. These compounds are metabolized by gut bacteria into urolithins, which have been studied for their effects on cellular health and mitochondrial function. Pomegranate also interacts with gut bacteria in ways that may influence immune balance, particularly species like Akkermansia mucinifila. This is where the connection between food, microbiome, and immune function becomes especially important. The gut does not operate in isolation. It communicates constantly with immune cells and influences systemic inflammation levels. What emerges from all of this research is not a single superfood solution, but a pattern. Foods that are rich in plant compounds tend to influence multiple biological pathways at once. They affect inflammation, oxidative stress, immune activity, and even gene expression. However, the real-world challenge is not identifying these foods, but maintaining consistency. Most studies that show benefits are based on regular, long-term intake rather than short-term changes. This means the goal is not perfection, but stability over time. Another important point is synergy. Foods rarely act through a single mechanism. For example, combining vitamin C-rich foods with other plant compounds can enhance absorption and effectiveness. Similarly, fats can improve the uptake of fat-soluble nutrients like lycopene. This is why dietary patterns tend to matter more than isolated foods. A combination of fruits, vegetables, legumes, and teas creates overlapping effects that reinforce each other. At this stage, the science becomes less about individual nutrients and more about systems. The body is not responding to one compound at a time, but to a continuous environment shaped by repeated dietary exposure. In the final part, we will bring everything together into a clear, practical framework. We will summarize the key foods, explain how to combine them realistically, and focus on how consistency and lifestyle patterns influence long-term cellular health and disease risk. At this point, the science comes together into a clearer picture. Cancer development is not driven by a single switch in the body, but by multiple systems interacting over time. DNA damage, immune surveillance, inflammation, microbiome balance, and angiogenesis all work together in a constant state of balance. When that balance holds, abnormal cells are usually detected and removed. When it breaks down, disease becomes more likely to progress. The important realization is that many of these systems are not fixed. They respond to the internal environment, and that environment is shaped daily by lifestyle choices, especially diet. This does not mean food alone determines outcomes, but it does mean food is one of the most consistent inputs the body receives. When we look at the research collectively, certain patterns repeat. Foods rich in polyphenols, flavonoids, and natural antioxidants tend to influence multiple protective pathways at once. They can support DNA repair, reduce oxidative stress, modulate inflammation, and in some cases influence angiogenesis, the process that controls blood vessel growth. The first key food group is kiwi fruit. Studies have shown that regular consumption is associated with reduced markers of DNA damage and improved repair activity. The effect appears linked to vitamin C and additional plant compounds that work together inside the cell. 1 to 3 kiwis daily has been studied as a practical intake range, with higher intake showing stronger effects in some cases. Green tea is another widely researched food. Its active compound, EGCG, has been studied for its interaction with pathways involved in blood vessel formation and cellular signaling. Regular intake, often around 2 to 3 cups per day in research contexts, has been associated with measurable biological effects. Preparation also matters, since temperature and steeping time influence the concentration of active compounds. Blueberries provide anthocyanins, compounds that influence both immune activity and inflammation regulation. Studies have shown that consistent intake can support immune cell function, particularly natural killer cells and T cells, which are involved in identifying and removing abnormal cells. 1 cup per day is commonly used in nutritional studies as a reference amount. Soy-based foods such as tofu, tempeh, and edamame contain isoflavones like genistein. These compounds have been studied for their interaction with hormone-related pathways and angiogenesis signaling. Population studies have observed lower rates of certain cancers in regions where soy consumption is consistently higher, although multiple lifestyle factors are involved. Broccoli, and especially broccoli sprouts, are rich in sulforaphane, a compound that activates detoxification enzymes in the body. These enzymes help process and remove potentially harmful compounds before they can damage cells. Broccoli sprouts contain significantly higher concentrations of sulforaphane compared to mature broccoli, making them a particularly dense source. Cooked tomatoes provide lycopene, a carotenoid that becomes more bioavailable after heating. Lycopene has been studied for its antioxidant properties and its role in protecting cells from oxidative stress. Regular intake of tomato-based foods, especially when combined with healthy fats like olive oil, has been associated in population studies with reduced risk of certain cancers. Pomegranate contains elagitannins, which are converted by gut bacteria into compounds that influence cellular and immune health. It also interacts with the gut microbiome, including bacteria such as Akkermansia mucinifila, which has been linked in research to stronger immune responses in certain clinical settings. When viewed together, these foods do not act as isolated interventions. They form a pattern of nutritional support that influences multiple biological systems simultaneously. The key point is not that any single food prevents disease, but that consistent dietary patterns can support the body's natural regulatory mechanisms. Another important concept is synergy. Nutrients often work better together than alone. For example, vitamin C can enhance the absorption of certain compounds, while dietary fats improve uptake of fat-soluble nutrients like lycopene. This means how foods are combined can be just as important as which foods are chosen. However, one of the most important findings from nutritional science is consistency. Biological systems respond more strongly to steady input than to occasional high doses. Eating a small amount of protective foods daily is more effective than consuming large amounts occasionally and then none for long periods. The body adapts to patterns, not isolated events. There is also a psychological component. Long-term adherence is more successful when the focus is on addition rather than restriction. Instead of removing foods entirely, building meals around supportive foods creates a more sustainable approach. Over time, this naturally shifts dietary balance without creating a sense of deprivation. It is also important to be realistic. Food is not a treatment for cancer, and it should never replace medical care. What the research suggests is that nutrition influences the internal environment in which disease develops. It can support systems that already exist in the body, including immune surveillance and cellular repair. Genetics play a role in risk, but they are not the only factor. Gene expression is influenced by environment, and diet is one of the most consistent environmental inputs over a lifetime. This means there is a meaningful degree of influence through daily habits, even if it is not absolute control. In the end, the most important takeaway is that the body is not passive. It is constantly repairing, defending, and regulating itself. These systems are active every second of the day. The question is whether they are being supported or overwhelmed. The foods discussed here are not shortcuts or guarantees. They are tools that interact with biology in measurable ways. When used consistently as part of a balanced lifestyle, they may help support the systems that keep cellular processes functioning properly. Understanding this shifts the perspective from fear to agency. Instead of viewing health as something determined only by genetics or chance, it becomes something influenced by daily choices over time. And that is where the real message lies. Not in eliminating risk completely, but in supporting the body's own ability to maintain balance as effectively as possible through consistent, evidence-informed habits.